Okay. Can you see the slide?
Yes, sir.
Okay, thank you. So, today I'm going to discuss about soil fertility evaluation. Actually, soil fertility evaluation means the diagnosis of nutrient status. This uh, and that means the level of nutrients in soil is the indicator of soil fertility. And actually, I have shown here two examples in fertile soil, which color is light, and another left side is rather dark color soil. Uh, this indicates uh, so there are several methods for soil fertility evaluation. One we can divide it in two major groups. One is chemical methods, and another one is biological methods. Under chemical methods, we can say soil analysis, which can be divided into two: total analysis and test for available nutrients. Another one is plant analysis, and under this plant analysis, uh, it can be divided into three: total plant analysis, plant tissue test, and biochemical method. And under chemical methods, there is another technique which is called nuclear technique. Now, biological methods. Under this method, it is also divided into two. Number one is use of higher plants. Under this, there are six techniques. Uh, Here, simple fertilizer trial on farmers' field, complex field experiments, methods of using plants as nutrient extractors. So you already, I'm not sure. Maybe some of you already some uh, extracting methods for nutrient analysis. For example, for phosphorus or potassium. So maybe some of you already did. Maybe some of you not yet performed. But you have to do that after opening the university in the laboratory. So that will be mm, good experience for you at the time. And also we can use indicator plants. Some plants may show specific symptoms on its plant parts for specific nutrients. And another one is visual diagnosis. This may be already in agronomy or some other classes. You already know some there are visual diagnosis you can do, you can perform uh, by looking the leaves or stems, and that can indicate the deficiency of nutrients. And some other technique is foliar spray. This is this kind of foliar spray. That means we can spray some nutrients on the leaves or stems. And there is another method, microbiological method. Actually, there are some microbes which we can use for uh, identifying the nutrient deficiency. For example, azotobacter plaque test, Aspergillus niger test, Kulinkula plaque test, carbon dioxide evaluation method. So, I already told you plant analysis under chemical test. Plant analysis, this method is the only way to confirm whether there is a deficiency or adequate plant nutrients are present in soil or not. However, these conditions may interrupt some conditions may interrupt nutrient uptake. For example, low soil temperature. If the soil temperature is low, then plant cannot uptake nutrient properly. The time if we analyze the plant, that may show the nutrients are inadequate, but actually nutrients are uh, present in soil. But how, in any case, uh, plant cannot uptake that one due to some adverse situation. For example, low soil temperature, very rapid plant growth, low soil water content, poor drainage, like this. So this is one technique, and now there is another technique. We have soil test, plant analysis, and soil test. Among these two, which one is better? If we want to make a comparison between these two, how can we conclude that one? Here you can see uh, plant is the final part where a plant nutrient is reaching, and although there might be plant nutrients in the soil, but may not reach in the plant part or plant stem. But in some cases, plant analysis is actually, this is the actual analysis, actual result can be found after analyzing the plant. But plant should be grown for analysis up to a certain period, and using the plant analysis means no pre-plan or fertilization during plant preparation. That means if you want to analyze the plant, then you have to grow the plant on the field or on the soil. So before so for this plant culture or plant preparation, you cannot prepare for fertilization, as you don't know the status of the field. And another one is further, you don't know the plant is deficient in an element until you see it or measure it. So you cannot say anything about the soil status. By that time, plant growth is reduced, and it may be too late to adequately correct the damage done to yields. For example, if you want to see the deficiency symptom on the plant, you have to grow it. But sometimes deficiency symptom, when deficiency symptom can be seen, that time plant growth can be reduced sufficiently, and that growth reduction reduces the plant yield. Only crops growing for a larger period of time may be suitable to predict fertilizer needs. This means if you have a crop growing for longer period, for example, for 1 year or more than or like that, that time you can correct the fertilizer needed. But if the crop duration is short, for example, 2 months or 3 months, the time you cannot correct the fertilizer after cultivation. Long duration crops are there are some example of long duration crops. For example, sugar cane. Even we can do this for a forest or other things. And another question sometimes we can ask, for example, total plant analysis versus quick tissue analysis. Which would be better or which should be performed? Actually, Plant analysis are accurate laboratory measurements of plant content of each element analyzed. This is the accurate measurement in the laboratory. In contrast, quick tissue tests are usually approximate measurements done on plant tissue. Quick tissue tests are done with some kind of chemicals and with plant strap. Plant strap means after crushing the cells, or for example, crushing the leaves or stem, we can get some extract that is called plant strap, and that can be analyzed for a short with a short technique. That's good for approximate measurement, but not that cannot make the accurate measurement. Scientists have decades thought quick, immediate measurements of nutrients in plants and soils. This interest in simple quick tests give birth to quick tissue tests. Unfortunately, quick tissue tests have been limited success because they're subject to operator errors and climatic change and are not quantitative. Mm, quick tissue tests, as I told you earlier, quick tissue tests have some errors. Maybe there are some, you can get some errors due to operator. If you are not expert in quick tissue analyzing, the time the result will be kind of error, and that will mislead you for fertilization. Usually, the current concentration of nutrients in quick tissue tests are reported only a very low medium. That is a qualitative test, not quantitative. Increasing ability to make total plant analysis quickly and easily has increased the use of plant analysis. That means increase total plant analysis can be done quickly for and this is using current days as one of the techniques. Quantitative value of any of the plant nutrients is possible within a day, but more usually within a few days. So this quantitative value, this for example, whether plant nutrients are low, medium, or high, this kind of test can be done within short time. However, among these points, quick tissue test is suitable for to adjust or to correct some problems. For example, irrigation, or we can consider what kind of fertilization technique should be done. That also we can do. We can consider from the quick tissue test. But however, for more accurate results, you must have to perform total plant analysis. So this is methods of plant analysis. Total plant analysis, plant tissue test, and biochemical methods. So now total analysis is based on the fact that an element deficient in soil would be present in plant in small amounts. So in view of shortage of one nutrient, the nutrients would tend to accumulate because of poor fertilization. So if poor fertilization, we do poor fertilization, the time nutrient shortage will be seen, we can see the nutrient shortage in the plants or plant parts, and plant analysis has been found useful. Hello, hello, plant analysis has been found. শোনা যাচ্ছে?
জি স্যার, জি স্যার।
Okay. Sometimes I may speak in Bengali so that you can understand it better. So not only in English, but sometimes I may speak in Bengali. Okay. So
Okay, sir.
now total analysis. So these are when we do total analysis, the time we have to test the leaves of same age, and with considering the level of yield. For example, we want to perform high yield or low yield or medium yield. That also should be considered during total analysis. So when what are the purposes of plant analysis? That means total plant analysis. When we want to diagnose or confirm visible symptoms, to identify hidden trouble or hidden hunger. I think you know already that is hidden hunger. Sometimes we cannot see any symptom in the plant part, but still there are some kind of shortage of nutrients, but cannot be seen in the leaves or other parts. That is called hidden hunger, and this kind of hidden hunger leads decrease of yield. To calculate in incipient deficiencies, to look at areas of incipient deficiencies, to identify the deficient areas and deficient areas, indicate whether applied nutrients entered the plant. That means although we are applying some kind of nutrients or fertilizers in the soil, but whether these are entering into the plant or not. To indicate interactions or antagonism among nutrients. This means interactions among the nutrients. For example, if there are some same kind of ions, for example, sodium and potassium, these both are cation, and these two interact between each other. One is there are some if sodium is high, then potassium cannot be uptaken. It inhibits the uptake of in any case. For example, calcium and copper. These two also interact each other. To aid the understanding of internal plant functioning. This means whether after uptaking the nutrients, whether that are functioning in in good way, and that also can be understood by total plant analysis. So now, how do we perform total plant analysis? We grow plants for 7 to 8 weeks in the field. Then we determine the total nutrient content for each nutrient. For example, separately we measure the nutrients, for example, for nitrogen or sodium, potassium, calcium, phosphorus. Each element should be measured in separate way. compare with the results obtained from the plants grown in standard soil. And these results after growing plants for 7 to 8 weeks, this is the actual soil, and another field will be prepared. We have to prepare and grow for 7 to 8 weeks. In similar way, if we do in nutrient deficient soil or actual soil for 7 weeks, you have to perform same way in standard soil. Standard soil means there should be all nutrients in standard condition. So these two can, after comparing these two results in soil and in standard soil, we can say this is someone is some nutrients are shortage, or some nutrients are excess, or some nutrients are in adequate condition. Another is a plant tissue test, which is called as rapid test and also qualitative test. That means this is not the actual measurement, but this is the qualitative test. Here, concentration of cell safe is usually a good indication, or how well the plant is supplied at the time of testing. আমরা যখন টেস্ট করব সেই সময়ে এটা কি পরিমাণ নিউট্রিয়েন্ট সেখানে সাপ্লাই করতে পেরেছে সেই বিষয়টাকে আমাদের নিশ্চিত করে, বা সেই বিষয়টাকে আমাদের ইন্ডিকেট করবে যদি আমরা প্ল্যান্ট সেফ বা সেল সেফ আমরা যদি অ্যানালাইজ করি কুইক টেস্ট হিসাবে আর কি, র্যাপিড টেস্ট বা সেই হিসাবে। The tender plant parts usually leaves are removed and plant sap is extracted. Okay, so we can make plant saps from leaves or special plant parts, then we can measure it through test. Well, actually, we perform this test for nitrate, phosphorus, potassium. Maybe you already performed this kind of test in the laboratory. No, not like this, but quick soil test. But there are some kind of technique to perform the quick test for nitrogen, phosphorus, and potassium for plant parts. The use of a specific reagent for each nutrient. This is a kind of this can be compared with the rapid soil test you performed in our laboratory. So, like that. So I would like to say something on simple field experiments on farmers field. In farmers field, you have to apply sorry, there should be one thing compulsory, that is control, and another one is nutrient treatment. Here in the for example table, you can see control, there is no fertilizer, and we can see the grain yield is 4 ton. This is just for example. And if we apply sulfur, then we can find the yield increases up to 4.8 ton per hectare. And if we apply only zinc, not sulfur, all others are same, that time it increases yield to 4.1 ton per hectare. If we apply sulfur and zinc together, that time yield increases up to 4.9 ton per hectare. Here you can see from control, if we apply sulfur, only sulfur, that time yield increases vigorously, But if we apply only zinc, the time yield increase is very little. And if we apply sulfur and zinc and compare with only sulfur treatment, the time yield increase also very little. So we can conclude from this table that uh, there is no deficiency of zinc, but there is a deficiency of sulfur. As after applying zinc, it's a very little increase. It's not or maybe zinc in the soil is medium, but there is a shortage of sulfur. And after applying sulfur, yield increases vigorously. And here is a example of complex field experiments on farmers field. Similar way as in previous. Here fertilizer rate for sulfur is 0, 15 and 30. And again zinc fertilizer rate is 0, 2 and 4 kg per hectare. So these two sulfur and zinc, sulfur is 3, sulfur is 3 and zinc is 3. So all are treated, all are all combinations will be made here. And obviously in this case also there would be a control. That means sulfur 0, zinc 0. This is the control. And here it appears that yield of rice increased considerably to 2 kg zinc. 2 kg zinc and 15 kg sulfur. So yield increases up to 4.9. This is the maximum yield. So this is the actual fertilizer dose. This will be the actual fertilizer dose for this kind of experiment. You can suggest after this experiment. Now, use of indicator plants. You know, there are some indicator plants you already studied in agronomy or some other classes. So I'm not going through this one. So there are some plants which can be used as the indicator plant. This is some visual diagnosis for nutrients. Example. But in this case, sometimes deficiency symptoms are similar with disease or chemicals or other stress effects. However, in the right side, this is the healthy plant of maize, and next one is deficient with phosphate, and third one is the deficient with potash, nitrogen, magnesium, and sixth one is drought stress. Seventh one is disease, and last one is some kind of chemical stress. So there are lots of nutrient deficient symptoms. You already these also you already studied in agronomy in the first year or other classes. So I'm not going through this. And foliar spray is one of the techniques to understand to identify the nutrient deficiency. Sometimes there are some kind of the deficiency symptom on plants, and if we spray specific nutrient for that, if after spraying that nutrient deficiency symptom disappears, then we can say this deficiency was due to that nutrient. For example, if you can see the nitrogen yellow leaves, Then the time we apply nitrogen as the spray, and after spraying that nitrogen, the deficiency symptom disappears. That time we can say this deficiency was due to nitrogen. Now I'm going to start soil test. Actually, you already performed this test from first year to present condition. Uh, there are some objectives of soil test. For example, if we want to make a fertilizer recommendation before crop cultivation, that time we need to perform soil test. Also, we can do probable crop response. We can predict probable crop response to applied nutrients, uh, and that also we can do other things too. This is the methods of soil testing. Rapid soil test we already and there are some rapid soil tests and uh, other tests also. And after doing this test, we can make a relationship of soil test and crop performance. Here are some advantages and disadvantages of soil test. I think these are not so difficult to understand. Uh, so I'm going to the next slide. These are the steps of soil testing and fertilizer recommendation. First step is soil sampling. We have to collect the soil samples from the field, then preparation and storage of soil samples. Step 3 is soil chemical analysis. Step 4 is soil test interpretation and field verification. Step 5 is fertilizer recommendation. And now I'm going to the soil sampling. Soil sampling, I don't need to describe this one, as you already did, you already performed soil sampling. How to collect soil samples from the field, you already performed. So this is you can remember from first year level 1 semester 1 classes. For field analysis, there are different kind of soil sampling technique. For example, one is field average sampling, which we do in the field like in the first year, but there are some other techniques too. For example, site-specific sampling. That is in case of heterogeneous and widely variable areas, the time we can take some sample from the uh, homogeneous soils. But if the soil changing to other kind, the time we can take samples in a good way. And also green sampling and directed sampling. That's I'm showing this sampling technique here in the field. For example, for field average sampling, left side you can see that is a zigzag condition, and 20 to 40 random samples should be taken from 10 acres of land. And grid sampling is done uh, like this way, after a specific interval, every specific interval. There are some, we have to select some spots after a specific interval, and that time we can take the sample from the from the source 2. In the right side, I showed one figure. Is This is the site-specific soil sampling. For example, if we have to collect soil samples from zone 1 that we take separately. We preserve it separately. Zone 2 from taking the soil samples from zone 2, that also we keep it separately and analyze separately. If we take samples from zone 3, that also analyzed separately, and fertilizer recommendation and other things also done separately. These are the consideration for sampling we already discussed in the level 1, semester 1. We take samples from the same depth. We cannot mix the samples collected from the different depths. And so these are the things. These are the time and frequency of sampling. Soil samples should be taken before seedling or actual going for cultivation, before going for cultivation. And every 2 to 3 years, we have to analyze the soil for fertilizer recommendation. And step 2, preparation and storage of soil sample. I am not going through this slide, as this already you did in your level 1, semester 1 classes. How to prepare a soil, for example, composite soil, making composite soil samples, sieving, and air drying, that all are included in this part. And leveling with information sheet. And next is the soil chemical analysis. This chemical analysis you have to perform. I'm not sure, maybe you did not start your soil chemical analysis in the laboratory, but however, you have to perform all this after the opening of the university. You have to do all this in the laboratory yourself, and that kind of analysis is part of final exam too. And next one is soil test interpretation and field verification. After analyzing, we can say whether some nutrients are shortage or not. আমরা যখন কোন একটা সয়েলের একটা কেমিক্যাল অ্যানালাইসিস এর পরে যে রিপোর্টটা পাই, যেমন সাপোজ আমরা সালফার পেলাম 10 ppm, সাপোজ we find sulfur is 10 ppm, but its critical condition is 14 ppm. That time we can say sulfur is deficient. Sulfur is 20 ppm, we can say this is sufficient. So that kind of interpretation is needed. And after that interpretation, we need to confirm with a field trial that is called field verification. Whether actually that shortage, that deficiency symptom was due to that nutrient or not. Sometimes some nutrient deficiency symptoms interact each other, and it looks similar, but actually that is not the cause. And finally, we make a fertilizer recommendation based on the yield goal. For example, we want to make a high yield, low yield, or medium yield. That also should be considered. In actual sense, we do 6 common interpretative categories. For example, very low, low, medium, optimum, high, and very high. These 6 categories are considered. That means আমাদের yield goalটা কি, আমার এটা আসলে it will depend on the farmer's ability. For example, if a farmer is very poor, he cannot input, he cannot contribute for input high yield, that time we can suggest him for a low yield. That means a low category of fertilizer. But if someone is using hybrid crop and with high yield goal, that time we can suggest him high fertilizer or optimum fertilizer for that crop or an crop and yield goal. So for micronutrients, there is a medium level of soil test value, and moderate yield goals has been assumed to be around 80% of high yield goal. That means moderate মানে আমাদের হাই yield গোলটা হচ্ছে আমাদের 100%, মডারেটটা হবে 80-র মতো, তারপরে তার নিচে আসলে 70%, এইভাবে আমরা কিন্তু এটাকে ধরে নেই আর কি। এখন আমি আসছি, now I'm coming to what are the factors affecting fertilizer recommendation. These 8 points should be considered. This can this 8 lead us to make a proper fertilizer recommendation. For example, soil, crop, and season. For example, soil is actually, you can understand whether if the soil is deficient in nutrients, that cannot be that can be corrected after applying the, but crop and season. For example, I already told you, if the some kind of native variety with low yield goal, that is different case from high yield goal and with hybrid crops. That means this also should be considered for the fertilizer recommendation. Nutrient level in the field, previous crop, if the some kind of residue of fertilizer from the previous crop is carrying, the time we can reduce fertilizer recommendation for the present crop. Desired yield, I already told you. Farmers' ability for management, methods of fertilizer application. This also important. If we apply in foliar or top dressing or side dressing, that methods of that also important. For production seed, this is also important. Whether we need to grow the crop for seed or vegetative part. If we want vegetative part, we need to apply nitrogen fertilizer more. But if we need seed, then we have to apply phosphorus or other nutrients more. Specific need for any crop also can be considered. For example, for mustard, we need to apply sulfur. That is a specific need for that crop. And now, why fertilizer may be in error? Although we do our test everything in good way, but there might be some problem which may lead us a error for fertilizer recommendation. আমরা যদিও এটা হয়তো আমাদের সমস্ত পরীক্ষায় আমরা সঠিকভাবে সম্পন্ন করার পরেও কখনো কখনো আমাদের এই ফার্টিলাইজার রিকমেন্ডেশনটা ভুল হতে পারে। সেটার কারণগুলো এখানে বলা হচ্ছে যে depth, time, composite of subsamples, that also may lead us for a error. Field history, that also a important thing. If there are some nutrients are carrying or shortage, or for example, if there are some residue phosphorus in the field, but we are applying phosphorus for the next crop in same way, that may occur a toxicity, toxicity for that nutrient. Actually, toxicity is not so frequently occur for macronutrient, but it may occur and frequently occurs in case of micronutrients. And number three is correction of laboratory test to field response. This is laboratory test for field response. I already told you in the soil test part that after analyzing the nutrients, we need to make a correlation with the field test, field response. Even though we analyze and predict in a good way, there might be some other things that may lead us a error in fertilizer recommendation. That needs a experience, your good thinking, oil thinking, and experience. These 2 points can rise this kind of errors. You have to you have to exercise your brain, your knowledge, and also experience. These two can minimize these kind of errors. And these are the points how to make a good recommendation. For making a good recommendation, we need a accurate cropping history. We need to know accurate cropping history, and this kind of cropping history we can get from the farmers only. If the farmers' information are misleading, that also that can make us wrong result. Laboratory must analyze the soil samples correctly. So that is very important. If we in person we are doing something wrong, that will happen. If the machine is not in good condition, that also make misleading information. So we should correct these two. Test evaluator must be trained in the job and know the area for which he makes predictions. So তার কাজ সম্পর্কে তাকে জানতে হবে এবং যে এলাকায় সে এটা প্রেডিক্ট করতে চাচ্ছে সেই এলাকা সম্পর্কে তার একটা ভালো আইডিয়া থাকতে হবে। এইগুলো হচ্ছে ফাইনালি দ্য রেকমেন্ডেশন ক্যানট বি অ্যাডভার্স ওয়েদার হেভি রেইন হেলথ মিনস শিলাবৃষ্টি যেটা ড্রট কোল্ড পিরিয়ডস পেস্ট ড্যামেজ পুয়ার ম্যানেজমেন্ট এগুলো যদি হয় সেটা ক্ষেত্রে আসলে ফার্টিলাইজার রিকমেন্ডেশন কোনো কাজে লাগবে না। এই হচ্ছে তোমাদের ফার্টিলাইজার সয়েল ফার্টিলিটি ইভালুয়েশন সম্পর্কে আলোচনা। তোমাদের একজন প্রথমে একটা প্রশ্ন করবে যদি তোমাদের কোনো প্রশ্ন থাকে। কোনো প্রশ্ন আছে? হ্যালো, হ্যালো, হ্যাঁ তোমাদের কোনো প্রশ্ন আছে?
মনে হয় কারোর প্রশ্ন নেই।
আচ্ছা আচ্ছা, তোমাদের প্রশ্ন নেই কিন্তু আমি উত্তর দিয়ে দেই। সেটা হচ্ছে তোমাদের এই হ্যান্ড আউটটা বোধহয় লাগবে। তোমরা কিভাবে এটা কালেক্ট করতে চাচ্ছ?
স্যার আপনি সিআর কে মেইল করে দিলেই হবে।
আচ্ছা ঠিক আছে।
সিআর আমাদেরকে গুগল ড্রাইভে আপলোড করে দেয় সব শীটগুলো।
আচ্ছা ঠিক আছে, I can do that very easily.
জি স্যার।
জি স্যার।
তো যাই হোক, আমার এই চ্যাপ্টারটা আমি আসলে শেষ করি। আমি যদি একটু বেশি সময় নিয়েছি, আমি তারপরেও এটা শেষ করতে পেরেছি। তা ভালো থাকো তোমরা সবাই। পরবর্তীতে হয়তো আরেকটা ক্লাস, আরেকটা চ্যাপ্টারে আর কি, অন্য আরেকটা চ্যাপ্টার লাগতে পারে। যদি লাগে আমি সেটা তখন